Hi guys, so this is Sam, and I'm going to be summarizing my lecture for you today. I'm going to be going quickly through it and just really pointing out the really important things that you guys need to keep in mind. So, you'll only really learn something if you didn't pay attention. So, population genetics. It is this field of study that studies genetic variation within gene pools and how that genetic variation changes. First, definition. A gene pool is the set of all alleles for every gene in a given population. Population geneticists pretty much study gene pools because a gene pool of a population is really the genetics of that population. But there's that word there, population. Well, there's another important definition in that because you need to be precise with your terms when you're a scientist. Population is a group of individuals, all of the same species, that all occupy the same environment and can all interbreed. These terms are necessary to make sure a population has a distinct gene pool. Examples of populations. There are the deer of New Jersey, bison of the plains, and if we all did interbreed, which we don't, the academy. Notice how these all are drastically different sizes and have drastically different sizes of environments in which they live. That's because a population really ends up being just like the group that you're studying that has a distinct gene pool. That's really how it's used. So the population doesn't have to be a specific size. You could study all the humans on Earth, or you could study just the humans of Bridgewater, New Jersey, as a population. So that is the definitions that we care about. Actually, I joked. There are more. Genetic variation. Well, there are pretty much two things that really matter in genetic variation. There are allele frequencies and there are genotype frequencies. Allele frequencies are the frequencies of some allele in the set of all alleles and in a population. And genotype frequencies are the frequencies of a genotype in individuals in a population. So that's cool. So there's this thing that talks about allele frequencies and genotype frequencies in stable populations, in which the gene pool is pretty much constant because there are no new mutations, no natural selection. It's big, so there's no genetic drift, and there's no migration messing up the nice frequencies of alleles that we have, and the mating is random. So populations of this sort settle into an equilibrium, which population geneticists like to call Hardy-Weinberg equilibrium. You should study this because it's probably on your test. This is the formula. This is the part you really care about. This pretty much says that p squared, so first of all, p and q are the frequencies of allele 1 and allele 2 of some gene, respectively. There are no other frequencies of that gene. There are no other alleles of that gene, although this model can be extended to accommodate that. So, what the Hardy-Weinberg equation says is that the frequency of homozygous p individuals is going to be p squared. The frequency of heterozygous individuals is going to be 2pq. The frequency of homozygous individuals is going to be q squared, and the sum of those is going to be equal to 1. This lets you really analyze populations, and you're probably going to be asked to do that at some point. So, when does Hardy-Weinberg work? Well, it works when there are no new mutations, so p and q don't change. There's no natural selection, so p and q don't change. The size is large, so there's no genetic drift, so p and q don't change. And there's no migration, so p and q don't change. It also has to be random mating, and that's for the whole derivation part to work, because pretty much the way Hardy-Weinberg is derived is that, the probability of having a p sperm fused with a p egg is going to be p squared, and that's the probability of you getting a p homozygous individual. Likewise, the probability of a q sperm and a p egg is pq. Probability of a p egg and a q sperm is pq. So the probability of a heterozygous individual is going to be 2pq. It's really not all that complicated. So that's how Hardy-Weinberg works. Now, so you can just remember what the point of Hardy-Weinberg is. It pretty much is trying to define a constant gene pool with random mating. And this is never actually true in reality because gene pools aren't really constant. Natural selection happens. There's pretty much migration. You can't really stop mutations. But although this is, never actually true, it models a lot of real-life populations really well. And yeah, that's actually how science works a lot of the time. It's nice. Approximate models that work surprisingly well. So that's static genetic variation. Now dynamic genetic variation. Populations can change. I know, right? It's crazy. So they can change in a size, location, or genetic makeup. So gene pools can change. They might get bigger or smaller. They might move. And that all these factors really affect each other, because if a population gets bigger, it might have to move to a new location, which moves it to a new environment, which causes new selective pressures on the population, which might in the end change its gene pool. Cool stuff. But the way this actually happens, like the particular way populations evolve, is called microevolution, and that's like how evolution works on a generation to generation level. So microevolution can either just introduce new alleles, make them from nothing. New mutations could occur, which would make a new allele. Gene duplication might occur, and then mutations might accumulate in the duplicated genes, making new alleles. Exon shuffling might occur, making new alleles, or new genes even. And horizontal gene transfer might occur, just like introducing genes from a totally other species into a species. And that's how you really make genetic variation. But most of evolution just has to do with fine-tuning the existing genetic variation. So natural selection is, you guys have heard of this so many times. It's just the process that really selects for increased frequencies of alleles that increase the fitness of an organism. So natural selection might select for instance, eagles being big and fast, thus being able to get more prey and have more offspring because they can have food during the lean winter months. That might help with alleles that contribute to bigness and fastness of eagles. There's also this thing called genetic drift, because there's a lot of randomness in this whole life thing. Sometimes individuals just die out because of an accident, and that can actually change your gene pool. Now, that only really matters if your population is small. If your population is large, all these little random events won't really be able to budge the frequencies of P and Q all too much. There's also migration, which is when eagles from Montana fly over to eagles from New Jersey and bring in new Montana genes from all the way over there into this population. And that can change things like genotypes and allele frequencies. And finally, there's non-random mating. Non-random mating is like, female eagles preferring to mate with male eagles that have, bigger claws. That might really help with the development of bigger claws pretty quickly, because you simply get the eagles with bigger claws will get more offspring, and thus alleles that contribute bigger claws will be selected for. So that is how microevolution works. And on a small scale, generation to generation, it causes changes, and over time it can cause really big changes, ending up in actual evolution and speciation and the whole thing that, creating diversity of life and all that. And so, just so you remember, population genetics is pretty much the study of evolution in action. That's all it is. It's really not all that bad. Good luck on the test, guys. I hope I was helpful. Thanks.